Alright, so this review is going to be mainly over it's going to be all over basically subjects from the test. So I went through the test and I looked at the questions that over 50% of the class got wrong. And I looked at the concepts of these questions, and so that's what I'll be reviewing. I'll have another review video where I'm literally going to go through every single thing into the nitty-gritty details of every single thing we need to learn for this test. But this one is just going to be all the questions that more than 50% of the class got wrong. So we will go through this. And this video is not going to be as in-depth as the one that I'll make tomorrow or Sunday. So basically we had the first one that more than 50% of people got wrong was this question right here. And the best way you're going to memorize this is to just fill in this table and study it. You got to know that stuff hydrophobic or amphipathic. You got to know amphipathic means it can be polar or non-polar. Like this lipid, which is what this is. The only one that stores information is your nucleic acid. The only one that can be a membrane transporter is a protein. So just learn how to recognize these things, and you'll be really good once you learn. We know that a polysaccharide or a monosaccharide, they look kind of like this, if you remember. So for A, when we're asked is this hydrophilic or hydrophobic, we see all this polarity. And we know that polar equals hydrophobic. Oh, sorry, not hydrophobic, hydrophilic. Sorry about that. Okay, so now we're moving to the next topic that over 50% of the class got wrong. Mind you, every single one I'm doing, every concept I'm going over is a concept that over 50% of the class got wrong. So this one, just straight up line drawings. So get used to these because these aren't going anywhere. These are here to stay. When you look at this drawing, you got to be able to know that every spot where this line bends is a carbon. There's not a carbon right there. And then you got to know that every carbon needs 4 things. Carbon needs 4, okay? So let's check this out. We're going to talk about this carbon. It's hooked to 1, 2. That's a bad color. Let me do a different one. Holy crap. All right. So this carbon right here has 1, 2. So we know it needs 2 more. So right there and right there. Boom. Okay, and we can go through this whole ring. We'll do this carbon right here, this one. It has 1, 2. We know it needs 3, 4. Okay, if you were to see this, okay, we got 1, 2. So we know there's 3, 4. If you were to see this, we have 1, 2, 3. We know it needs a 4th. So for over here, we got 1. So we know it needs 2, 3, 4, and these are hydrogens. If you want to get good at this, you have to sit down and every other night do 50 of them. It's super easy to draw out 50, and it's pretty quick to go through all 50. Yeah, it's not going to be fun, but getting good at this stuff, it's just not fun. You're never going to have fun studying this stuff. You just got to do it. The fun comes after. Okay, so all right, so we covered line drawings. Now we're going to go over the strength of bonds. So there's two types of bonds. There's covalent and there's non-covalent. Non-covalent, there's ionic, H bond, and van der Waals. For covalent, we have polar, and nonpolar, okay? So, which one is stronger than which? And I asked this, you guys were asked the question, you have this protein and it has all these bonds in it. Which bond is going to require the most energy to break up? This right here is the concept that that question was testing. So you have to know that a covalent bond is stronger than a non-covalent bond. You just got to know that. That's going to be important for chemistry. It's going to be important for maybe biochem later on. And then when we get down here, know that an ionic bond is stronger than a hydrogen bond, and a hydrogen bond is stronger than a van der Waal. As far as polar and nonpolar, they're about equal. So no, covalent is stronger than non-covalent. And then over here, ionic is stronger than hydrogen bond, and that's stronger than a van der Waals. So the next topic was hypertonic and hypotonic. And this one can get tricky, so I totally understand. We'll draw, hopefully after this, you are good to go. So this is our cell. Just doing its thing, hanging out. Hypo. And so these are, we'll say it's a salt concentration. Oh, 1 second, because I'm having a little brain fart too. One second. Okay, cool. So on our hypotonic, okay, so I was doing it. Then hypertonic. Okay, so we're going to say that we're all doctors, and we have this patient come in. In this patient, let me finish drawing this. So, zoom this out. All right, so this cell is going to turn to that, and this cell is going to turn to that. So why does this matter? Why would you even want to do this? And why does this even happen? So the reason, we'll talk about this one first. The reason a hypotonic solution is going to expand a cell is because the cell has a higher concentration of salt or whatever inside of it, and the water follows the salt, right? So this cell is hypertonic compared to the solution. This hypotonic is literally just explaining the ratio of the solution around the cell to the cell. So there's less salt around the cell than there is inside. Therefore, the exterior or the solution is hypotonic. Over here, we have a lot more solute outside of the cell than we do inside, and therefore the solution is hypertonic. So let's say your patient, because you're a doctor now, has tons of swelling. Let's say they have congestive heart failure and there's tons of swelling in their lower legs. And we want to get rid of that. So as a doctor, you'll probably prescribe some sort of hypertonic IV solution. And that's going to be like a short-term remedy to bring the water out of that tissue. How in the world is it going to bring the water out the tissue? Well, since it's a hypertonic solution, it's probably going to be high in some sort of salt or minerals or something. And when all that stuff goes by, it's going to, all the water that's in this cell is going to leave out the cell and bind with all this stuff. And it's going to shrink up the cell. Where, I don't know a case where you'd want to give a patient a hypotonic solution. We'll just say their cells, or they're going through some sort of cellular atrophy. And we want these cells to expand and absorb more water. Maybe in dehydration this happens, or a hangover or something. So we're going to give them a hypotonic solution, which will basically be a very dilute saline concentration. I know most IVs are hypotonic, and it's just like a 0.9% solution of sodium chloride. That's your standard IV bag, is that, which is a super small percentage. And that's going to make the cell expand and take on more fluid. It's going to have a higher fluid retention. So if what I just explained wasn't good enough to really make this sink in, and this took me a long time to learn too. I still don't even have it memorized, obviously. Study it on your own. You got to just hit the books hard. Keep hitting the books hard, and you'll get there. Sometimes things don't just click right away. You got to draw it forward and backwards and forward. Just do everything you can, and it'll click eventually. So the next thing is active transport. Cool. So there's active and there's passive, or diffusion, whatever you want to call it. There's primary active and there's secondary. And then over passive, we have diffusion, which is passive, and then we have facilitated. Holy cow, I spelled that wrong. Facilitated. Okay. So a primary active transport is going to use energy. That's the main thing you need to know. Use energy. And a lot of the time it'll be you'll see as the word pump. An example is the sodium-potassium pump, which helps bring resting membrane potential back to normal. So active is always going to be low to high concentration. Passive is high to low concentration. A secondary active transporter is so a secondary active transport uses the energy potential, the kinetic energy from a spontaneous reaction to power a non-spontaneous reaction. So let's draw that out so we can visualize what the heck I'm talking about. This is spontaneous. This is non-spontaneous. This is a symporter. So we can see we have a spontaneous powering a non-spontaneous. I'm just going to write non-spon. This is also, in some areas it's called reaction coupling or coupling. It's when you have something that's non-spontaneous, but in order to make it happen, you couple the reaction with something that is so spontaneous it has extra energy to drive the reaction you want. Okay, so diffusion, it's going to be things like O2 H2 into all your gases, small hydrophobic molecules, and then facilitated is what you're going to need for your big polar stuff. So like sugar or something like that, you'll need facilitated. So this is active and passive transport. Know the difference between these? It's pretty important. It's definitely important for this class, and it won't be the first time you guys have to know this for a test. So just get it down now and build a big foundation for the rest of your college career. And the last concept, what is attracted to the backbone of DNA? So I talked about test taking skills a few times. All you needed to know for this question is what the backbone of DNA looks like, and you could have gone to your monomer, macromolecule. There's a question that showed like all sorts of random bits and pieces of macromolecules and monomers, and you would have seen something that looks like this. And this is a phosphate, and it's also the backbone of a DNA. And if we notice up here, we have a negative charge, and we know that a positive is attracted to a negative. We got negative and we got positive. There we go. They like each other and they like to be around each other. But we know this is negative, but what is positive? And the options were like an acid or a base. Well, if you went to your table that shows all the amino acids, the answer is right there. So flip around in your book, and this is why I always like to go through tests 2 to 3 times. Because the first time through, let's say you don't know number 5, but then maybe like number 22 might actually have key information for solving test 5 or question 5. But let's say you're on question like number 28 and you have no idea what it is. Go through your test a second time and you'll find supporting information that can help you answer that. So go through your test multiple times. Hit the books super hard. If you didn't get the score you want, just sacrifice this weekend. Hit the books every single day, and you'll get the score you want. So that's it for this video. I'm gonna post a really long video Saturday or Sunday, and I'll try to make links in the video so you guys can just skip straight to what you want. I'll try to make it easy for you. But yeah, that's it for this video. So just keep hitting the books hard, and see you next week.